Yes, hello. Good morning. So, Hank, yeah, you asked about Punnett Squares, and this is a topic that frequently comes up. So I thought I'd make a video about it just to go through this in more detail. I have another video that I did in another course that I can link you as well, but this one will be a bit more specific to the way it's covered in Science 3106. It's a topic that comes up a few different times. But before I get into sort of the nuts and the bolts of how Punnett squares work, I wanted to just talk a little bit about how they were developed. So, first up, um, the study of genetics basically began with an Austrian monk by the name of Gregor Mendel, and Mendel bred pea plants that the monks used, I guess, for food sources or whatever. And he noticed something very curious while breeding his pea plants. And these pictures I'm going to bring up here are from the a different biology textbook than the one you're using, so you won't have seen these, but it's going to be on the exact same topic that you're dealing with. So over here on the side now, we can see P. You see a P, an F1, and an F2 down along the sides, and those are terms that tell us what generation it is. P meaning the parent or the first generation. F means filial, but we just need to know that that's sort of the children generations, right? So F1 is the first generation of children. F2 is the second generation of children, right? So you breed P, the first two, to get the F1 generation. Then you breed the F1 generation to get the F2 generation. And in doing so, right, in basically crossing pea plants and then breeding them with other ones that have been resulted from the same cross, Mendel noticed a very distinct pattern, and it's not something that you would necessarily expect from the start. He noticed that if he crossed a short pea plant and a tall pea plant, he would usually end up with all tall pea plants as the offspring. It's quite odd, right? You'd think that if one parent plant was short, one parent plant was tall, then the offspring would be probably in between somewhere. But that's not at all what he found. What he found was that usually, or a lot of the time, every single one of them ended up tall. But then if you took those plants that came from that one and then crossed them again, what he ended up with was a 3-to-1 ratio, as we see here toward the bottom. Now, it doesn't mean that it was exactly four plants, obviously. This is, 75%. It's a 3-to-1 ratio, right? 75% of them would come out tall, about 25% of them would come out short. And this happened again and again and again as he did more and more trials of this. So, this is sort of what began the study of genetics and passing traits through what we call dominant and recessive genes. Now, if you went on and did more biology, and in the course that this one is from, it does go on and it talks about there's other types of dominance like incomplete dominance, codominance. We're not going to be looking at those in this course. It's not that much more complicated, but for now, we're just going to be looking at this straightforward example that's exactly like what Mendel found. And then to keep on going, so yeah, when he crossed a tall and a short, like I said, he ended up with all tall plants. Um, just to skip ahead a bit, I guess, this the explanation for this can be found through our Punnett squares, right? There are things called alleles. Alleles are these letters that we're representing here. And so a tall plant, if we were to take so alleles are the letters that we use to represent this, right? So a capital T here represents a tall gene. A lowercase t like this represents a short. Now each organism will have two genes that govern these traits. Okay? So the capital T is tall and the lowercase t is short. And this is what's called the genotype. Actually, I'll get rid of this picture for now so we can see my paper a bit better. So these are called genotypes. And that means the letters used to represent the gene. So when we have the possible genotypes, there's 3 different ones. Okay, you don't necessarily need to memorize these terms. I will use these terms sometimes when I'm explaining it to you, but 2 capital T's like this is what we call homozygous dominant. Homo meaning same, and zygous is just the phrase we use to mean the zygote, basically, right? These are the genes. Anyway, homozygous, 2 of them are the same, and they're dominant because they're both the tall gene. And then we have this one. This is what we call heterozygous. And 2 lowercase t's is called homozygous recessive. Okay, so these are all the possible genotypes. And now phenotype is what Mendel was observing. Phenotype is how the trait is displayed, I guess. Like what we end up actually seeing, right? And for TT, the plant ended up being, I'll call this possible phenotypes here. What we find is that with 2 capital T's, it ends up being tall. With 2 lowercase t's, it ends up being short. But if there's 1 of each, it ends up being tall anyway. Okay, and this is really the core of what Mendel started to find. And if I go back to that diagram that I only recently brought up, if we have a tall plant that is homozygous dominant, right, 2 capital T's, like we have in the top left there, then we breed that with a short plant. For the plant to be short, it has to be 2 lowercase t's. Every combination that we get is going to be an uppercase T and a lowercase t. And how do we know that? Well, we know that by representing it through this, right? This is where the Punnett Square comes in, because this shows us the possible crosses that we can have. So let me go ahead and do, I'll keep doing it in different colors down through here. I'll show you now how we would get one like this, and then I'll do ones for different combinations. Then I have a practice activity that I'll show you how to work down through. So it's a little bit different than the one that you're using, but either way. So now when we're filling in a Punnett Square like the one we see here, like you see on the top, we write one of the different parents. So for this first cross, right, Mendel's, so we're talking again, right, about Mendel's first cross over here, the short pea plant and the tall pea plant together. So in doing that, the tall pea plant, I'm going to put its 2 alleles here. The short pea plant, I'm going to put its 2 alleles down here along the side. So this is one parent, this is the other parent. And now in each of these squares, I'm going to put the letter from the vertical and the letter from the horizontal. And it's convention to, if there's a small one, to put it second. I wouldn't mark you wrong for putting the small one first. Just, it's worth knowing that it is convention that if there's a small one, it'll go second. So that's what we end up with, right? We have one from the vertical, one from the horizontal in each of these squares. It's just like, it's like we're multiplying sets of numbers. This is kind of how it works as well, right? But what we notice is that if one parent is homozygous dominant, which is this, one parent is homozygous recessive, which is this, all the children plants, they all end up being heterozygous, or one of each. Right? And again, if we look back to Mendel's cross, we see that in the F1 generation, they all ended up being tall. Right? So F1 generation, I'll say genotype is 4TT. So it's all heterozygous. And the phenotype will be 4 tall. All of them are going to be tall. But now what would happen if we crossed F1 in order to get the F2 generation, which is the next part down? Now, the graphic to the left there does tell us what it's going to be, but let's walk through why using a Punnett square. So, we're crossing two of the F1 generation, and all of the F1 generation are heterozygous. So we're going to put one heterozygous along the top, like so, and one heterozygous along the left, like so. Now, like I was mentioning, we're going to take the vertical letter and the horizontal letter and write them in each of these blocks. So here it's going to be 2 capital T's. Here it's going to be a capital and a lowercase. Here it's going to be a capital and a lowercase. And here it's going to be 2 lowercase t's. So now this is what we call the F2 generation, or the second filial generation. The genotypes. Now here we're going to have a more significant ratio here. There's going to be more things in it. We're going to have 1 heterozygous dominant to 2, homozygous dominant, sorry, to 2 heterozygous to 1 homozygous recessive. Right, just like that. And then our phenotype. What's our phenotypic ratio going to be? What are the different phenotypes? Remember, from up here, right, 2 capital T's or 1 capital T, 1 small t, they all end up being tall. So this and this are tall, and this one is short. So we have 3 tall to 1 short. Okay, and that's what we end up with. This is why Mendel observed this this pattern. And him seeing this pattern again and again and again over all the different times that he crossed this led him to realize that there must be something going on other than just, you know, the children are obviously not just an average of what the parents' genes are. There's something more sophisticated happening. And this led him to the discovery of basically how the modern study of genetics kind of began with Gregor Mendel and his pea plants. Sorry. Now, to carry on here, like I said, I'm going to go through a few examples. This might be a little more accurate to the way that you're using Punnett squares in your activity. Now, in your activity, you are doing all the different combinations. So, actually, yeah, before I move on. Oh, sorry. Didn't mean to hit the microphone. My goodness. Look at that. Poor sound design. So to show you all the possible combinations, I'll just write this over here. Now we don't bother with which one's first and which one's second, right? Whether it's first or second doesn't matter, because this is, you know, I actually teach math mostly and not science mostly, strangely enough. But for St. Anthony, I'm teaching only the science. But this is like matrix multiplication. And in multiplication, we know it's commutative, which is a fancy way of saying the order doesn't matter. So we're not going to be concerned with, say, if we had, I'll do this on a separate piece of paper because I'm not going to include this with the notes that I send you. But if we had, say, TT crossed with TT, or like these two ones are the same. So we wouldn't consider these to be two different crosses because the order of which one is first and which one is second doesn't matter. But our possible combinations are going to be homozygous dominant by homozygous dominant, homozygous dominant by heterozygous, homozygous dominant by homozygous recessive, And now since the order doesn't matter, we don't need to do this one again. So we can have heterozygous by heterozygous. We can have heterozygous by homozygous recessive. And then we can have recessive by recessive. So when your activity tells you to do all 6 crosses, these are the 6. Okay, so it's dominant by dominant heterozygous, dominant by recessive, heterozygous by heterozygous, heterozygous by recessive, and then recessive by recessive. So those are the different combinations that you are going to be doing in that activity. Again, I'm going to now show, and I will send you these notes. Actually, well, I don't even need to say will, because by the time you get the link to this video, you will also have the notes. So you have the notes. Yeah, this is, and I'll send you the this practice activity as well. It's quite a good one. And I'll go through a few of the questions here. You can do the rest of them yourself if you would like to have some more practice on this topic. But like it says at the top of the screen there, a type of flower shape is determined by a single gene in which the dominant version C creates a circular flower, recessive version lowercase c creates a square flower. What is the likelihood of a circular flower if plants of the following genotypes are crossed? It is worth saying right now, something I didn't mention, that we always use the letter for the dominant trait. That's just a convention, right? You'll notice here that it's a capital C and a lowercase c. Even though it's square, it's not a capital C for circular and a lowercase s. We use the same letter both times to show that these are the genes that are dominant with each other. That's not terribly relevant for what we're doing right now, but if you went on and did a little bit more in this topic with the incomplete dominance, then it would start becoming more important to follow with the naming conventions. So we will stick with that, right? That's why back over here, we didn't use T and S. We used capital T and little t. So then this is going to be 1A here. So we've got heterozygous and then homozygous recessive. So let's go through and draw out a quick Punnett square for those. So again, we do one parent across the top. It doesn't matter which because the order doesn't matter. And then the other parent down this side. Then out in the middle here, we're going to combine capital C, small c, small c, small c, capital C, small c, and small c, small c. So, what is the likelihood of a circular flower? So since we're looking for a circular flower, we know we're looking for the phenotypes. Right, so here our phenotype is going to be 2 circular to 2 square. Right, because we have our genotypes is 2 heterozygous to 2 homozygous recessive. And in some things, like say, when we talk about people being, say, a carrier for a disease that sometimes but you don't have the disease yourself, that's a different type of dominance, but it's kind of like this, right? You can imagine this person, say, or this one would be a carrier but wouldn't have the disease, whereas this one would have it, you could say. And you see that, like, if a carrier would have children with someone who had it, then there would be a 50% chance to have it if this is the way it works. Okay, that might be a little bit confusing. We'll scratch that for now. But you would see that sort of stuff talked about if you went on to do more about dominant and recessive genes like so. But here anyway now, we have half of them are circular, half of them are square. So what is the likelihood? Likelihood of circular is going to be 2 out of 4, because it's 2 here out of 4 in total, which is, of course, 50%. So 50% is the answer there. And to show the answer, I would be looking for a Punnett square like so. Let me scroll down to, yeah, let's do this next one. So B is looking for going to do homozygous dominant and then homozygous recessive. Now, since they're both the same, I think you can maybe predict already what we're going to get, because this is the same as Mendel's first cross. All right, so we've got a homozygous dominant, a homozygous recessive, and we're going to see that we're going to have a capital C and a small c, capital C and a small c, capital C and a small c in each of these. So our genotypes is going to be all heterozygous. Our phenotype is going to be 4 circular. So likelihood of circular is going to be 4 out of 4 or 100%. 100, 100, 100. There we go. Running out of space on my paper. You know, I'll leave the next 2 questions on here. They're pretty good questions, so if you would like to have some more review, perhaps, of Punnett squares, of how they work, you're more than welcome to do questions 2 and 3 on this sheet and submit those as well. Right? That can perhaps help show you the way these work. I hope that was a decent introduction there. Again, right, the combinations that you are going to be doing are going to be all 6 of these. Now, I've done one similar to a lot of those, but I think you'll find that once you sort of understand the way to set the squares up, I think it'll be fairly straightforward for those. Okay, so I hope that helps. Let me know if you have any other questions about Punnett squares.